Hey kids, welcome back to biology. Today we're covering the Unit 3 Human Digestion Lab. I'm doing this for you as a lab help to help you get over any problems you may have getting everything started. So you will need the Human Digestion lesson notes. This is the teacher copy that I have here that you can see, but you're going to need to have your student copy out and available. So pause the video until you have that open. Now that you're back, the Human Digestion Lab is, before you can get started doing that, we want you to understand a little bit of the behind the scenes in digestion. So that's why I've provided this reading for you that's sort of interspersed between the intro and what we actually did in the lab. So take a few minutes and read these paragraphs when you download your own. It talks about how humans digest their food and how it's a physical and chemical process and it's a multi-step process. And in class, if you remember, we accessed this link here, which when I click on it, brings us to this. And so when we play this, it walks us through, I turned the sound off. It walks us through all the descriptions of the parts of the human digestive system, and it walks through all the parts. So
into smaller particles during mastication. Salivary glands located near the oral cavity secrete saliva, which begins chemical digestion and keeps.
But you get the idea. So I asked you to watch this link here so that you can prep yourself, take some notes on this in this section here of the guided of this student version of the notes. Okay, so make sure you do that. If there's something in here in this 1 through 9 that you don't feel is properly covered, there's also a section at the end of your notes here that has a keywords and pronunciation section, as well as some more explanation for those parts and pieces. So if you don't find it in the video, feel free to look through that. You need to be prepared to take a quiz. The quiz you'd find here, if you did it, would be at the end. Where's it at? Oh, it's down here. So you can practice the quiz here if you want. I'm recreating these quizzes as a warm-up Friday when you come into class. I would like you to be able to do this with us in a polling section with a good educated guess for what we're talking about, all right? So make sure that you do that on your spare time. You have Beacon Buddies tomorrow, so you should have time to do that. And then we were talking about getting into the lab right after that. So I've provided you with some more reading here because this is the part from the content where if you went to the content, your home page, if you went to the content and you went to Unit 3, this year it's Module 10 and 11. So if you went to Unit 3, you'd see there we go. If you go to 310, you'd find you'd find the intro, like you would see in all the other lessons. You'd find the intro and the explore parts. I just consolidated those for you, and so you'll find that in the 310 and 11 lesson notes student copy. Oh, that should say student. I've got a typo. But that's what we're looking at over here. So when you download that, that's what this is we're looking at. And you have parts that you need to fill out here because I'm asking you to write them down so that you understand what's happening in the lab. Too often we get started in a lab, we have no idea what the point is. So in this lab, we're going to simulate digestion. We're going to take an egg white, which is mostly protein, and we're going to put it in different solutions that you would find in your own human digestive system. So we're going to put it with hydrochloric acid, which is what you find in your stomach, as well as an enzyme called pepsin. So those two things work in your stomach to break down, for example, the egg or whatever you're eating into smaller pieces. And then the chemical nature of what's happening in your stomach is that it is incredibly acidic. It's very the whole purpose of it is to break materials down. So if we just let that go through our digestive system without neutralizing it, it would eat holes in the rest of our digestive system, our small and our large intestines. So we have to add sodium bicarbonate at some point, which you'd see here in this explanation. And sodium bicarbonate acts like Tums or Alka-Seltzer does if you have a tummy ache. If you have a tummy ache and your stomach's all gurgly and it hurts, it's probably because you have too much stomach acid. So the bicarbonate neutralizes it because it's a base. So you'll learn about more of that in chemistry. But if you have an acid solution and you combine it with a base, you end up with water and a salt. So that's why we would do that, so that when it goes through the rest of your intestinal system, it doesn't burn a hole in your gut. All right. Then we're also going to look at the digestion of milk. So we're looking at digesting two things in this lab: a protein in the form of an egg white and milk. Now, milk has proteins in it, but we're going to be looking at it to find the sugars, because that's the other thing your body breaks down to use besides protein, and that's glucose, right? So milk is a combination of glucose and galactose. So we're going to break that milk sugar down to get to the glucose. So you need to understand that lactase is the enzyme that we use to do that in our body, unless you're lactose intolerant, right? And then you have a problem breaking those down. And then we're going to be looking for the presence of glucose after the fact. Then the other thing we're going to be investigating is the properties of human digestion, right? So we're doing that in a virtual setting. We're doing that so that we find out how the body chemistry changes when food is added, how the food is broken down, and what the process of digestion is. That's reinforcing what we're learning today that we had in our objectives from up here in the top, where it says, can you describe the process of digestion in a human, citing examples from anatomy? And do you know the individual functions and sites of secretion of digestive enzymes, right? So where does the HCl, the hydrochloric acid in your stomach, come from? Where does that bicarbonate or bile come from, etc.? And then I've set you up with the labs and procedures that you need. Now you can find them also here in your lab report. Now, if you're in my 11:45 session, you'll choose the S1. If you're in my 12:30 session, you'll have S2, session 1 and session 2. So you'll find those when you open those up. They're going to have the same steps that you see here in what I've crafted for you below, more or less. There's the difference is section 1 is doing a discussion and section 2 is not. So it's going to be just a little bit different in that regard. But that's what you're going to turn into me. So when you turn in your homework, you're going to turn in one of these two depending on which section you're in, and that'll be on Friday. All right, and then let's get started in part A for the procedure. It says follow the directions to conduct the lab. We're going to set it up today, but we have to have 48 hours before everything gels out. So we're going to start it, and then we're going to get a code at the end of D, section D today, and we're going to write that down. Alrighty, so follow the procedure to conduct the lab. So the first part is to open the digestive virtual lab, which you find in your student version. And I'll just click that here, and that's this. I'll put it here just for me right now. Okay, so you should find that in your student version already. And if not, I'll update it so it for sure does. I think it does. I just don't think it has it on mine. All right, we're going to click the tutorial button first. We're going to choose our gloves. I like blue. I don't know why. I just always choose blue. Hit start. And then it's going to walk us through the process. Click on the gear, click on the arrows to move things around. Okay, what I want you to notice, I'm going to get this off the screen real quick. Click on the resize icon, that's this. Click on the volume, click on the zoom to make things bigger or smaller. Resize icon, I did that already. Lock to lock the aspect ratio, I don't like to do that. Click on the speaker on or off. And then drag the bar toward the magnifying glass to make things bigger or smaller. That's zooming into the room, okay? Click on the Home tab to reset the lab.
Click on the hard-boiled egg to prepare it.
Okay, well hold on before we do that. I want you to notice on the wall over here is some indicators for us. This is a pH chart. Remember I said how we're dealing with acids and bases? So you're going to take one of these strips at some point, and you're going to have to test your solutions and compare it to this chart here. And you're going to take some of these strips when you're dealing with the milk, and you're going to compare it to this color scheme here. So when it asks you to do that, you want to come over here to the wall to do that. So the narrator is asking us to click on the hard-boiled egg to prepare it. Well, I don't know where that is, so I'm going to zoom out. Oh, there's the egg, right? If you want to just get a view around the room, here's a timer, stirring rods, there's the liquids we're using on the top shelf, graduated cylinder for measuring stuff. This is a pipette way of measuring stuff. Then we have two racks of test tubes. This is the one we're going to use for the milk. All right, so this is going to be part B, and this is the one we're going to use for part C. So right now we're only doing part A. We're doing the introduction over here, and it's asking us just to complete the tutorial. All right. Now, if you look down here where it talks about egg, we're going to have to redo all of that. So since I'm doing it for you, I'm going to have you pause this video, open yours up, and do your tutorial. Make sure you're comfortable with what you have to do. But I'm not going to do the tutorial since I'm going to end up doing it all over again anyway, and I already know how to do it. All right, so here we are back at the main page. I went to the home button and reset the lab so we could start from here. So when you begin the lab for part B, because that's what we're on, is part B, we've already done the tutorial. We're going to start the lab at part B. Pick another pair of gloves, white. Okay, so let's see what we have to do. For part B, it says milk digestion. So we're going to be focusing on the milk stuff today. Now remember, today, both days, but now, we've got two test tube setups here. One here is for milk. That's for part B. That's where we're at. And this over here is for part C because that says protein. All right, so this is what we're going to be working with, and the milk because, we got to do that. So this says, using the pipette labeled milk, place one pipette full of milk in each of the 4 test tubes on the rack. All right, so if we don't know what a pipette is, let's look at what our options are for measuring in the lab. We have this beaker. I could pick it up, but it's not going to let me put it in. I can't pour it because those aren't measured for volume, okay? Those are just for mixing. So I need something that's going to measure. Well, this, if I pick this up, this is a graduated cylinder. And if I look really closely at that, I could measure out 2.5 mils. Like worst case you did that, you wouldn't be wrong to measure out 2.5 milliliters here. But if you did that, you'd find to pick up the milk pipette, you want to use the one that's labeled milk so you don't cross-contaminate. If you picked up the milk pipette and you go up to grab some milk and put it down here, sorry, if you go up to the milk, and you go and put it in here, you're going to find that that's 2.5 mils. So let's just test, right? So if you go here and look at this again, well, that's going to read 2.5 milliliters on my graduated cylinder. 1, 2, and some change. Okay, so that's not a really good way of measuring it. So the pipettes are, it says, use the pipette labeled milk to place 1 each in there, all right? And then in number 2, it says, fill each test tube with 2.5 mils or 1 dropper full of each liquid as indicated in the table. All right, so let's see. So we're going to dump this because we don't need it. Go, 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 go. Why can't I move? Oh, I have my whole screen off. Here we go. And I just rinsed it, okay? So it says, use the pipette labeled milk. Place 1 pipette full of milk in each of the test tubes. So that's 1, 2, 4. Okay, then you put that down. Put down, put it down, put it down. All right. Fill each test tube with 2.5 mils or 1 dropper full of each liquid as indicated on the table. Well, where do we get the table from? That's over here. Okay, so liquid 1, milk, is in all 4 already. 1, 2, 3, 4, that's done. Check. And then it says liquid 2. We need to add water, hydrochloric acid, lactase, and lactase. So, let's see. Let's zoom into this area a little bit. All right, so where do we get those materials? Well, here's hydrochloric acid. Here's pepsin. We don't need pepsin this time. Lactase and sodium bicarbonate. So we need this one and this one and water. So we're going to use those. So this pipette here says water. So the first one, we're supposed to take water and put it in the one that says milk plus water. Okay, and then we can put that back. And then this one, it says, for number 2, it says hydrochloric acid. So let's put hydrochloric acid in. That back. And then the 3rd and 4th ask for lactase. So I'm going to put lactase in the 3rd, and I'm going to put lactase in the 4th. Okay, so that's the second column for liquid 2. Then for liquid 3, you don't need to put any extra liquids in 1 through 3, but you need hydrochloric acid in the 4th one. So I'm going to do that real quickly. Okay, and then I put that back. All right, and then it says when milk is digested, I'm reading over here under the table, it says when milk is digested, lactose, a sugar, is split. Releases glucose and galactose. The test strip indicates the presence of sugar. Do not use these strips yet. What strips, Mrs. McInally? Well, those are over here. So after, when it tells you to use the strips to check to see if glucose is present, you're going to use these strips, and then you're going to dip it in, and then you can compare it to the chart. But it says don't do that yet, because it's not done digesting. It needs a little time. It says write your predictions about what will happen with regard to the presence of glucose after the test is finished in the milk digestion data table. Well, that's down here if you scroll down. So you need to write a prediction here. So I'm going to say write your prediction here. What do you think will happen? Okay, if you have hydrochloric acid, oops, my bad, milk and water. Let's zoom in here so we know what we're saying. So the first test tube is milk plus water. All right, so what do you think would happen? We're looking to see if there's any digestion happening. So write your prediction over here on the table if you think there's going to be any digestion. And if you think so, well, then we'll write the results later. So write your prediction. You'd say the milk was digested or the milk was not digested. And again, if you want to know what you might be looking for, here's that table. If glucose is present, in other words, if the milk was digested to break down into glucose, then you'd see a yellow show up when you used this strip on there later. If you didn't, you would see that it stays a light colored blue here. If it's somewhere in the middle, then some degree of digestion happened. Okay, so no digestion, and it would be light blue. Some digestion, or yes, it dissolved, it digested everything, and now there's glucose present, and you definitely see yellow. Those are your options. So let's go back down to the other test tubes. Okay, milk and acid would be what you'd write for number 2. So for tube 1. And then here you'd say, you write your prediction. Write your prediction. Tube 2, then 3, write your prediction for milk and lactase. Now, what is lactase again? It talks about that up here in the reading. What does it say about lactase? I know it said something. So let's read through here again, where it says, oh, look, here it is. It says, you will also study the digestion of milk. Lactase is an enzyme that breaks down lactose into glucose and galactose. You will test a digested milk sample for the presence of glucose. So, if lactase is present, that's what breaks down the milk into glucose. All right, so at that point, you would have to predict, with glucose present here, will the milk break down, right? And what level it was. So you'd write your prediction here. I don't need to write this prediction for you. You guys need to be able to do this without me. I'm just showing you what to do and where to do it. All right, and then the last one is not just milk and acid, but it's milk and lactase and acid, right? So that's probably going to be significant, right? Write your prediction for tube 4. Okay, so we've done that part. We're going to come back up here. Pause the video here and make sure you have a good scientific description. Use a color. I predict I will see blue. I predict I will see yellow. I predict the glucose will be completely dissolved. The glucose will be partly dissolved. The glucose will not be dissolved. That's the kind of thing I'm looking for in your predictions. Pause if you need to. Okay, then it says that's done. We're done with part B. That's it. All right, so that's all we do for now. We'll look at the results later. Part C is protein digestion. So you're going to test your protein. That's the egg. Remember, if you've ever heard of bodybuilders, if you've ever heard the expression that bodybuilders eat a lot of eggs, oh, that's ugly. Don't do that. That's because there's a lot of protein in eggs, and bodybuilders like to have a lot of protein in their diet so that they build big muscles. Oh my goodness, this is making me nauseous. Let's get this a little smaller. Sorry about that, guys. I know I'm neurotic about the little things. All right, so we gotta go to the egg. So what does it say for part C? Mince the egg. This is your protein. Well, where's an egg in this whole thing? Let's see. Oh, there it is. So I'm gonna pick this spoon up, or no, I'll pick the egg up. Oh, I guess you just have to like touch it a lot. I've never minced an egg with my fingers before, but hey. All right, so C1 is done. C2, using the spoon, place 1 spoonful of egg white in each of the 6 tubes. So 1. And it would have walked you through how to do this in the tutorial, by the way. Because remember, I'm in this section now because I'm working with the protein. You wouldn't mix the protein and the milk together. That would be confusing. You would be testing 2 different things. So keep your results clean and not cross-contaminating anything. All right, and then it says fill each test tube with the solution as indicated in the table. So let's look at the table for protein. Test number 1, 10 mils of water. Okay, so now we can use the bigger graduated cylinder. So I'm going to pick this up, and I'm going to pour. Can't pour it into here, bummer. What should we use? What do you think we should use to get the water into the beaker? Or the cylinder, I should say. Well, there's a handy dandy little pipette here that has water written on it. Let's try that. So 10 mils, that's 2.5. That's 5. 7.5. Well, that's a pot. And that's 10. All right, so that's 10. I'm going to take this, and I'm going to add it to the first one. Okay, now do I have to rinse this if I had water in it? What am I rinsing it with? Water. So you don't have to rinse when you're using water, but when you start with other things like pepsin, then you're going to need to rinse between, okay? Because you don't want to cross-contaminate. So the second tube says to put 10 mils of pepsin. So that's 1, 2, come on, there you go. 4. All right, put that back. All right, then this goes in the second tube. All right, now I have to rinse this because I've contaminated it with pepsin, and I don't want to put pepsin in my next one because that's supposed to have hydrochloric acid. So I take hydrochloric acid, and it looks like 10 mils. Now, I'm going to make a little shortcut here, all right? Just bear with me. I just put 4 of these into this graduated cylinder in order to make 10 mils that I could later pour into here. So I'm wondering if it'll let me take a shortcut and put 10 in here. So that's 1. No. 1. Okay, good. Count with me. 2, 3, 4. All right, and we're going to put that back. All right, then it says 10 mils of sodium bicarbonate in the fourth one. Sodium bicarbonate. Okay, the fourth one gets sodium bicarbonate. Let's just double check because we're kind of far away. So let's make sure we're doing what we're supposed to do and putting what we're supposed to put in. So put pepsin, acid, and base. The base is bicarbonate, remember, because it talks about that up here. So bicarbonate talks about that up here where it talks about here. It talks about that here. So let's do this. Green, right? So here you go. This is where it talks about the base, and the base would be bicarbonate in this case. All right, so sodium bicarbonate. I'm going to put 4 in that. 1, 2, 3, 4. All right. Now in the lab, I wouldn't want you to put your pipette right in there, so I'm kind of surprised it did that. But notice how full these are here in just a second, okay? Then the next one, 5, pepsin and sodium bicarbonate. Okay, so I've got to do 2 pepsin in the 5th tube. I've got to do 2 of these. And 2 of what now? 2 of sodium bicarbonate. So 2 of that is the green tube. Okay. All right, we'll put that back. And then it says 5 mils of pepsin and 5 mils of hydrochloric acid. So I'm going to take the pepsin and 1 more because I want 5 of each. So that's 2 times 2.5. And the hydrochloric acid, that's over here. Okay. And one more time. Okay. Now let's just talk about what we did here for a second, all right? Let's bring a little bit of chemistry into it. Now, in our description of what's supposed to happen with the protein, okay, it says 2 substances we'll use are hydrochloric acid and enzyme occur naturally in your stomach, and they both break down proteins. So the hydrochloric acid and the pepsin both break down proteins. So here they are. This one and this one are what you would naturally find in your stomach. They are good at breaking proteins down. All right, and then it says sodium bicarbonate, which is this one, neutralizes the actions of these 2. So keeping that in mind, okay, that would make these 2 not reactive, okay, because it neutralizes them. And then lactase would be irrelevant. We didn't even put it in the protein, so we don't have to talk about that. So let's talk about our test tubes here. In the first one, we've got egg whites and water. So you could do this right now. Go get an egg white, boil it, and then float it in water and see if it digests. Okay? And then this one is protein and pepsin. Now we know pepsin does break down proteins because it says it right over here, right? It says it right here. I'm going to use an even different color here. I'm going to use the blue. All right? I don't want to do that one, but it's fine. Okay, so this has some ability to digest. This one has an acid, hydrochloric acid, which also digests proteins. This one has a protein and a base, so that neutralizes any kind of digestion there might be. But a base might react with a protein. This is the protein plus the pepsin plus the base. So remember up here it says the base, sodium bicarbonate, is going to neutralize the actions of anything that's digesting your stomach acids, like the pepsin and the acid. And then in this last one, it's got the protein plus the pepsin plus the acid. Okay? So we're going to make some predictions about what's going to happen in these tubes, and they're going to ask you which of these is going to digest the most and which is going to digest the least, and etc. There'll be some levels of digestion going on in them. So let's see what it says. says using the forceps, pick a pH test strip and determine the pH for each protein test tube. Oh, and we have to record it in our results. So now we get to use those pH strips. So just to see if you remember, the pH strips are over here. These are the pH strips. These are the glucose strips. Use these ones for the milk, these ones for the protein. So we're going to grab one of these. Oh, forceps. That's what I got to use these. Can't use my fingers. You don't want to use your fingers to use for these test strips because your fingers have acid on them. All right, so I'm going to test this one. What does it say? It says, okay, compare it. So that's 7. So we would write 7 down in our, where do I put this? Is there a trash? Put down. Wow, what do I do with it? Uh-oh, I'm stuck with it. Well, let go. How about that? Okay, well, I'm going to go over here to my data table. Let's see. Oh, still holding on to it. So the test strip results. Okay. What does it say? Record in the protein digestion data table. Huh. Okay. So we need another column here. You guys can just write it next to it. All right. So pH strip results. All right. So this test tube was green, and it was pH 7. Okay. No, I'm still holding on to this thing, and I don't know how to let go of it. Is there trash somewhere? That's nuts. Okay, I don't want that. Let go of it. Huh. Well, that's fun. Okay, well, at this point, I guess what you're going to do is you're going to finish this part out. All right, kiddos, I figured it out. You just have to stick it to the wall on the pH strip. Sorry. So I figured it out. Sorry it took me so long. It's been a year since I did this one. So, all right, and then the next one, you take another pH strip. You bring it over here, and you dip it in. Seems premature. Okay, and that one is looks like 7 to me. Let's try another one. Why is it picking up green? Oh, this one's got something. Here we go. That goes with, what do you think? Let's zoom in. And come over here, and we'll compare it. Let's see. Looks like that one to me. Or maybe that one, but that one. This one should be 7 as well. Doesn't want me to put it there. It must be 8. All right. This one. Okay. So this fourth one, 1, 2, 3, 4 is blue. I might zoom out again. Okay, the fourth one is blue. Let's see. Let's zoom in. Now I think that's not placed right, so I'm going to pick it up and I'm going to move it over here because it looks like it fits better here. All right, pick another one. Fifth one. Wait, blew past it. 1, 2, 3, 4, 5. Ah. Looks like it's still green. It's much closer to 8 this time. All right. So it's 1, 2, 3, 4, 5. Let's write these down before we forget. The second one was green. pH is 7. The third one was red, orange. And that was a pH 2. The fourth one was blue, purple. We'll say blue. pH 11. Oopsie. Okay. So now we have to leave one more. And we need to go to the last one. Oh. All right, the last one was orange again. Orangey. More orange than red, I would say. Okay. Or maybe. Yeah, more red. Yeah, more like that. Okay. So I would say this one here is going to be closer to, the pH 2 is more like 2 or 3. So I'm going to say 2.5. And this one's definitely closer to 1. Oh, I forgot this other green one. This one here is our other one. So this one should be probably a little bit baser. Yeah. So I would say these two are 7 and this one's closer to 8. All right, so we forgot this one is green, pH 8, and this one is red, pH 1.5, probably. We're going to split hairs. So, yeah, it's definitely like 1.5. I'll say 1. Okay. So it says, we're back up here to part C, number 5. We've done number 4. It says, in the predictions column, write a prediction about which solution you think will digest the egg most completely. All right, so we have to look up here. So remember what we talked about, what's in your stomach normally and how well you digest an egg. So you have to think about that in terms of pH. Acids and pepsin are going to have a low pH. The lower the pH, the more acidic the solution is. The higher the pH, the more basic or not acidic, right? So both of those are pretty extreme ends of the pH scale. Like if you put bleach and stuff, you're going to make some changes. So any extremes might be noted, but at this point, if it's neutral or close to water, you probably wouldn't find a lot of change. But you're going to write your prediction here for test tube 1 through 6. Oops, I hate that. Okay, that's what we're there for. That's C5. Explain your reasoning. So make sure that you put that in your prediction. In fact, you could say, with explanation. Okay, don't forget to add an explanation. In other words, why do you say that? One good thing you might want to put in there is the color that you've seen on the test tube strip. And what does that mean? Okay, so keep that in mind. Okay, very good. And what? We're on D. Set the timer. So now we're done with what we have to do, and we're at what? How long? It took us 40 minutes. We could have done that in class more or less. Oh, I gotta put this down. Get it done. All right. So then it says place a stopper on top of all the test tubes. Okay, well these are stoppers. So these little black things are stoppers. They're supposed to stop anything from happening, any contamination from getting into the stopper. So give it to me. Okay. Give it to me. Oh, so frustrating. Here we go. Put it. Puts the cap on the test tube. There you go. Wow, this is taking longer than anything else. Okay. Woo! All right, then it says, once all the test tubes have been covered, click start on the timer. Well, don't you have to put time on the timer first? It says we have to wait an entire 48 hours. After 48 hours have passed, enter the code in the space provided on the opening screen. There we go. It says 48 hours, right? Okay, so we're going to do this. We're going to take this. Give it to me. All of it? Oh my goodness, seriously? Give it all to me. Okay, I'll just add that. Looks like an I. Yeah, that's an I. Okay. And I'm going to put it over here. I'm going to put it right here. And I'm going to make it big. And I'm going to make it bold. And I'm going to make it red. Or what other color do we like? I'm going to make it green. Yeah. And I'm going to hit V. Oh, come on. I just asked it to do all that. All right. Bold, underline, italicize, and do that. What the heck? Can't miss that, right? All right. I don't know if there's supposed to be an I, so I'm going to put a space and add one just in case, because I'm not sure if it just didn't copy or not. So looks like there's an I, right? So we'll just see. All right, and then hit OK. And then I believe we're done. What does it say? For the reactions to take place, wait an entire 48 hours. Enter the code in the space provided in the opening screen. The program will bring you back to the lab where reaction will have taken place. And then we do our observation. So we'll do that on Friday. So let's zoom out. Say goodnight to our test tubes. Bye-bye, test tubes. I'll see you in 2 days. I'm going to hit. Okay, kiddos. So when you get to that screen that I just flashed through when you click out, it's going to say, do you want to reset your data or not? I chose not to do that because I was worried that would start me from scratch. So I just clicked the X button like here, and I just clicked out like this. I did not choose to reset my data. So please be aware when you do that, I would just click out. Just exit out like the normal way, okay? So then in 48 hours when we come back, we should be able to access that and see what we've got. All right, I will see you on Friday. In the meantime, happy labbing. Bye.